All the attending members are requested to please stay connected. Okay. So, a good morning to one and all present here online for the inaugural function of the short-term training program organized by Ballarpur Institute of Technology. With great pleasure, we welcome you all to be a part of this inaugural function. My name is Abhijit Tehadrai, and I am your host for this short-term training program. Once again, on behalf of Ballarpur Institute of Technology and on behalf of the organizing committee of this short-term training program, I welcome you with all my heart. Respected chief guest for this inaugural function, Honorable Mrs. Vanshika Adiani ma'am, all the respected faculty members and enthusiastic participants of this short-term training program, Honorable Patron of our institute, Mr. Sanjay Vasade sir, Director of Ballarpur Institute of Technology, Dr. Rajnikant sir, Coordinator of this faculty development program, Dr. Archana Nimkar ma'am, and co-coordinator of this short-term training program, Mr. Neeraj Singh Baes sir, and organizing committee members of this short-term training program. It is indeed a great joy to have you all for the inaugural event of this short-term training program. It is to our observation very rightly said that by learning you will teach and by teaching you will learn. So is our experience also. Furthermore, in today's ever-changing and evolving globally connected world, learning is a practice that cannot be compromised with, especially so by the teachers, lecturers and professors. Again, as rightly said, who dares to teach must never cease to learn. So I congratulate All India Council for Technical Education for engaging in this continuous endeavor of learning and providing such learning opportunities in the form of short-term training program to our fraternity to grow faster and better. Dear attendees, we would like to inform you that this is the third batch of the short-term training program, also commonly addressed as STTP. It starts today. It is a 1 week STTP which is being conducted and which will be conducted from December 21st to December 27th of this year 2020. As you are aware, this short-term training program is sponsored by All India Council for Technical Education, that is AICTE. The topic of this STTP is biotechnology and its application in tribal agriculture areas. We would also like to inform you that the first and second batch of this short-term training program was conducted from November 23rd to November 28th and December 7th to December 12th of this year 2020. Further, with immense pleasure and pride, we state that it was a parcel of interesting facts and in-depth knowledge sharing clubbed with exclusive experiences of our guest speakers. So we are now starting to celebrate learning in batch 3 of this short-term training program. I repose a lot of confidence in you that all the respected attendees of this program will make it a huge success with their presence, participation, interaction and experience sharing. So let us all convert this short-term training program in a lifetime learning experience. Once again, I welcome you all to attend and participate in the third batch of this AICTE sponsored 1 week short-term training program. Now may I request the coordinator of this short-term training program, Dr. Archana Nimkar ma'am, to share her opening remarks. Over to you, ma'am. Ma'am, I think you are on mute, Archana ma'am.
Yes. Am I audible now?
Yes, ma'am, loud and clear.
Good morning to all my dear participants and honorable dignitary. It's matter of pride and immense pleasure for us to organize an AICT sponsored 1 week short-term training program on biotechnology and its application in tribal agricultural areas. Also, I would like to extend my warmest welcome to all the participants across Maharashtra who actively shown their keen interest in online learning. My friends, no one thought of that situation that we remain at home for such a long duration. Here comes new opportunity to explore opportunity of online learning in our educational sector. Whenever I heard the word biotechnology, I always remember this simple statement. There where is the life, there is DNA, and where there is DNA, There is a biotechnology. According to me, a new technological revolution in industrial and agricultural sectors in the next decade will be substantially and beneficially influenced by biotechnology. There is a great potential for improving the agricultural productivity by modifying plants, animals, and microorganisms and developing newer techniques like vaccine, tissue culture, genetic engineering, etc. The purpose of this 1 week short term training program is to spread information about various biotechnological advancements and develop aware commoners, especially in tribal areas, by providing a common platform to biotechnologists, industrial managers, researchers, and professors. The main objective of the STTP program is to provide online virtual training and make all the individuals aware about various cutting edge technology used in agriculture, innovative methods, and hybrid skill mechanism that will be required for agricultural development in tribal areas. The online STT program will be delivered by eminent resource person, speakers from well-known universities, institutes, and industries. We all have an opportunity to prepare ourselves for better tomorrow. I wish you all a fruitful and unforgettable experience in a coming few days of this online STTP as you explore the e-learning and virtual classroom education. I hope that you will learn some of new things from this STTP. Let us all meticulously follow the advisory and precautionary measures laid down by the respective authorities and win the fight against coronavirus. Stay home, stay safe. I welcome you all and I hope the 6 days STTP program will be a lifetime learning experience for all of you. Thank you very much for joining us and I welcome Vanshika ma'am for accepting our invitation to guide us on this occasion. Over to you, Abhijit.
Thank you, ma'am. And thank you very much for your opening remarks, ma'am. And now we take great pride in introducing to you our honorable guest for the inaugural function of third batch of this short-term training program. Our chief guest for the inaugural function is Mrs. Vanshika Adiani, ma'am. She is scientific officer in Food Technology Division of Bhabha Atomic Research Center, Mumbai. She has completed MSc in Biotechnology from Rashtrasant Tukadoji Maharaj Nagpur University. She has completed BSc in Microbiology, Biochemistry and Chemistry. She is also a gold medalist in BSc Chemistry from Rashtrasant Tukadoji Maharaj Nagpur University. She has qualified GATE exam in Life Sciences in the year 2007. Furthermore, she has also qualified CSIR UGC NET exam in Life Sciences in the same year 2007. She has published few book chapters and several papers in renowned peer-reviewed international journals. Her research interests include natural products chemistry, volatile organic compounds and essential oils, food irradiations, predictive microbiology, chemometrics and data mining, time temperature indicators, food adulterants, and contaminants. Mrs. Vanshika Adiani, ma'am, it is a matter of great honor and pride for us to have you as our chief guest in this batch 3 of All India Council for Technical Education sponsored short-term training program on biotechnology and its application in tribal agriculture areas. May I please request you, ma'am, to please address the gathering now.
A very good morning to all of you. It is indeed a great pride to be part of this training program, to attend this inaugural session. I'm really grateful that I'm now the part of this show, and belonging to Balarpur, I really feel it is a proud moment for me to connect with my roots because I have been one amongst you. So I'm really very happy to be here now. Um, I will like to share my slides.
Yes, sure, ma'am.
me share can you see my slide?
Ma'am, right now the slides are not visible.
Okay.
At the bottom of the screen, ma'am, you can see that there is a tab that says present now at the bottom of the screen. If you take cursor,
Yeah.
you have to click on present now and then
Okay.
you can share your entire screen. So whatever is on your
Sure. Yeah.
screen, that we will be able to see them.
So is it okay?
Yes, ma'am. Now we can see your presentation.
Okay. So as you know that I'm a food technologist now. I started my journey being a biotechnologist. I did my MSc in biotechnology, but I ended up being a food technologist now. So a little out of way, but definitely I feel that you'll be able to make some sense as to these kind of technology also may be utilized for biotechnology applications, because now this the topic which I'm going to talk about, it is very multidisciplinary. So I really feel that you would be able to take out some essence of the whole work, of the whole work which I'm going to present. And moreover, I'll also present about radiation processing, which is a basic work which we do at Bhabha Atomic Research Center, because it is an organization where radiation applications are being explored. So I'll start my presentation. So why at all we require to study food quality? The very first obvious choice is it gives a successful competitive advantage in market. It is like whenever we go into food vendor shop, we also are always looking for a product which is really good in quality. We have our own preferable food vendors where we feel that the quality of the product is really good. So this is an obvious choice that food quality is very important characteristic. And the second characteristic is we should maintain the quality of food products so that we also prevent the food losses. We know that there's a huge global burden of food spoilage throughout the world. It is not just in India, but food spoilage is a major problem throughout the world. So according to FAO, that is Food Agriculture Association, about 1.3 billion tons of the food gets wasted every year. That is about 1/3 of the product which we have produced gets wasted. Uh, these are the different type of food product varieties, that is cereals, where we see that 30% of foods gets wasted, 20% in dairy, 35% in fish products, 45%, tremendous amount of wastage in fruits and vegetables, which we also usually see in APMC markets or around us or in food processing industries. So 20% of meat products gets wasted. Similarly, oil seeds and pulses, 20% wasted. 40% in roots and tubers. So this is a huge amount of food that gets wasted. So if we see about in case of fruits and vegetable as an example, this wastage occurs at different processing levels, like at harvesting, producer level, storage level, transport, then at wholesale markets or retail stores. So there's a huge amount of food products that gets wasted. So it is not important that now we think about producing. Now we have a surplus amount of agricultural produce that is being produced in agricultural sector. The need of the hour is that we should now stop this food loss, food spoilage. Also, this food spoilage leads to a lot of loss in water, land resources get wasted, energy, electricity gets wasted, the labor and the capital, the amount of money we are investing gets wasted. So this also leads to a lot of greenhouse gas emissions, later leads to global warming and climate change. So even Government of India is taking a lot of initiatives through Ministry of Food Processing Industries so that we avoid this wastage of agriculture produce. Now coming back to the food quality, if we see scientifically how we determine this food quality, these are determined by several of the parameters, like in case of fruits specifically, if I talk about, we measure soluble solid content. This parameter will tell you about how ripe, how juicy the fruit is. Firmness also gives us the quality of freshness of the food. Then pH also plays a good role in determining the juiciness and the ripeness status of the fruits. Color also gives the parameter. I mean, seeing the color also we get attracted whether the food quality is good or not. Then tenderness, in case of meat products, it is important how tender the product is. And chlorophyll content, in case of green leafies, it is a very important criterion which scientifically is being studied. So microbial quality is also another important factor which is specifically being studied in case of fresh cut produce. Nowadays, industry is growing. We can see several fresh cut fruits that have been available in the supermarkets, and they are now in trend because now they have provided a lot of convenience to the customer. They don't have to cut, peel, and the product is ready. They just have to consume it. Okay. So in such kind of products, microbial quality estimation is the important factor. So if you see how we measure the quality of these products, traditionally, some of the techniques I have given are oven drying to determine the moisture content, colorimeter that is required to estimate the color. These are all scientific parameters that we use to study the product quality. Then texture analyzer, this is some specific instrument that will give you the texture. How is the mouth feel of the product? Okay, and then the conventional plate count technique you all must be aware, which we use to determine the microbial quality estimation in the food product, which is obviously a very important choice because it also ensures food safety. So these, if you see this traditional analytical techniques, these are generally expensive. They are very slow, and then they require analytical skills. Like a person who is having these analytical skills only will be able to handle these instruments. So if you see these techniques, they are generally destructive. Like they cannot be used in a food processing industry where a product has been developed and online monitoring cannot be done for these kind of products. So we require a technique which can be suitable for online monitoring in the food processing industry. So there is a need for a development of non-contact, non-destructive online technologies that can be used in the industry. So imagine an industry, for example, maybe a ketchup industry. So there, if the ketchup is being produced, if you want to see whether the color of the ketchup is good, whether the quality is retained, so you cannot go for this kind of traditional techniques. You require that there is just a click shots. For example, a camera just taking a photograph, and you get to know the color. Yeah, it is perfectly okay, and then the whole lot is being passed. You have to imagine this way. Such kind of technologies are required by the industry. Okay, so now many industrial approaches have been utilized in the industry, wherein different analytical tools that have been utilized, like optical techniques, UV visible, near infrared, then mid infrared, Raman spectroscopy, fluorescence, hyperspectral imaging. Hyperspectral imaging is a trending technology now these days because it will take the photograph. I mean, the pixel images also gives. Moreover, it will give multispectral information about the product. So such kind of optical techniques will allow us to directly determine what is the sugar content of the food product, what is the acidity, what is the color, what are the, if there is any internal defects. So such kind of parameters can be directly studied by using this technique. So you do not have to do a destructive processing of the product and then monitor the product. Then the mechanical techniques such as acoustic sound and ultrasound, they will determine the stiffness, texture, and degree of maturity. So again, imagine a watermelon. Okay, you cannot just see the watermelon and tell what is the quality of the product, whether it is really ripe or not. We usually ask the vendor, okay, whether this will be a ripe product or not. So such kind of techniques will help us to directly determine whether the product is ripe and it is in optimum condition for the consumer. Okay, for the juice processing industry, it is really a very good technology because they don't have to manually process and then get to know whether the product is good or not. They can identify online whether the watermelon is in a correct ripe stage and can be utilized for making a juice. Okay, so other chemical techniques such as GCM, this is gas chromatography, mass spectrometry, liquid chromatography, mass spectrometry, and ENOS. They are also utilized to determine the acidity, sugar, H-based volatiles, Okay, through using these techniques, we can get to know what is the food quality status of the product. Okay, to appreciate what I'm talking about, I'll highlight about how my work has gone into this field and how I have developed my techniques to online monitor the microbial quality of minimally processed fruits using different instrumental techniques. Okay, am I very fast or is it okay?
It's okay, ma'am.
Okay, so it is understandable, right? Or should I go a little slow?
No, it's okay. Understandable.
Okay, okay. So basically, I was working on this minimally processed fruits. You can see an image where pomegranate, jackfruit, and pineapple has been processed. Okay, so nowadays you can see such kind of packages that are available in the market. So these are called as minimally processed fruits because herein you have just physically altered the original form. You have just peeled, you have cut, you have obtained the fruits the way they are consumed, and you have packed it. So they really allow a good convenience. Imagine a big jackfruit and you're bringing it home and processing it. It's really difficult. Many of them will not prefer to consume these products because this is tedious to consume. I mean, you have to really go out of like you have to put a lot of labor to process such kind of products. So such kind of products have found a boom in industry where these are processed and these are available in supermarkets. So such kind of products, the beginning of these products started with hotel and catering services because they found that if they could get such kind of product easily, it will be an easy hand for them to perform their operations later. Then later, they are now also available in quick service restaurants, specifically in developed nations, and supermarkets in India and retail stores as well. You may have even seen that sprouted noodles that are available. So they have got a recent huge boom, such kind of products. And the future for these products are, you may see a vending machine where you can directly just take out the product and you can consume it. So that will be a good prospect for school and workplaces where you would better prefer for a healthy snack than going for a not so healthy options. So the advantages these product gives us, they are easy in serving portions. Of course, like a single person can consume the whole product at a time. They reduce the cost of they extend the shelf life because you do some kind of modified packaging, so that will also ensure the shelf life of the product remains long for a longer time. And then it also provides a selection choice for a consumer, so the consumer can directly see and make a decision whether it is good to consume or not. Sorry. So there are different kind of products that are now being minimally consumed in the uh, throughout the world, and I have provided the overview of the products that are being minimally processed. Now I'll be specifically talking about the microbial quality evaluation. Now you see this product is open, so this is vulnerable to microbial contamination because the nutritive surface is already available outside. I mean, the microorganisms can keep on increasing their counts, and so they become a bigger threat for such kind of products. So it is very important that the microbial counts of these kind of products has to be uh, well kept into count before determining the shelf life of the product. I mean, on shelf, if we say that the shelf life of this product is for 3 days or 4 days, so there is a scientific research that has to be conducted. You know that how long the product's count will not go greater than 7 log CFU for the total viable counts. These are the FSSAI limits that are being set, and the yeast and mold count that is said to be less than 4 log CFU counts. Okay, so we all know how to do this conventional plate count technique, that is the standard plate count technique, and we also know that to get the exact counts will take 48 to 72 hours for the count. Now imagine a product which is having a shelf life of only 3 to 4 days. How can we wait for 48 to 72 hours to determine the microbial count? Of course, we cannot wait. So we require a really fast technique which can tell what are the log counts of this product. And moreover, these standard plate count technique is labor intensive. You have to do serial dilutions, then plating, So, and it also requires a skill handling person who can do it. And this is a destructive technique. So there is a need for a rapid monitoring methods of microbial quality assessment in industry, which are preferably non-destructive. That is, you do not have to degrade the product. Online, the product is passing and something on the computer is displayed and it is telling that, okay, now the counts are somewhere around 3 log CFP. Okay, it is good to consume, it is fresh. So such kind of technology is required by the industry. Okay, so mostly for this food quality estimation, the organism that have been targeted are total viable counts. These are basically the total viable bacterial counts, which is given in CFU per gram, that is colony forming units per gram. Or we may go for a yeast and mold count, or we may go for pseudomonas. These are the primary spoilers mostly in fruits and vegetables. Enterobacteriaceae, wherein we have used bad quality water, you will find E. coli, Salmonella. And then specific organisms, for example, if you are going for a juice, you may have to determine Alicyclobacillus or Listeria. Or if the product is in saccharotropic conditions, then Bacillus thermospata. So that completely depends upon the food you are going to target. So those kind of organisms have to be studied. Okay, if you see the overall work that has to be performed is, we know that this plate count technique is very cumbersome. It takes a lot of time, that is 48 to 72 hours. So the alternative techniques wherein we are using some instruments that can be utilized directly by the industry. In such kind of this technology has for this technology that has to be developed. We need to use
Am I audible? If somebody will please confirm.
Yes, you are audible.
Okay. All the attendees are requested to stay connected with us. I think there is some connectivity issue because of which we are not able to hear our honorable resource person. We'll get in touch with them and resolve the issue immediately. Until then, please stay connected with us. Thank you.
हो आता राहील गुरुवारपर्यंत
All the attending members are requested to please stay connected with us. I think we have lost our we have lost connection with our honorable speaker, because of which we are not able to hear us, because of which we are not able to hear her. You are all requested to please stay connected with us. We are trying to establish contact with her, and we will resume the session soon. Until then, please stay connected. Thank you. All the attending members are requested to please stay connected with us. We are trying to establish contact with our honorable resource person. Because due to some connectivity issue, she is not able to join us in the meeting. You are all requested to please stay tuned until then. Thank you. All the attending members are requested to please stay connected with us. We have lost the contact with our honorable resource person due to some connectivity issue. She is not able to join in. We will request all of you to please stay tuned with us. We will try and resolve this issue soon. Until then, please stay connected with us. Thank you. Due to some technical difficulties, our honorable guest, our honorable chief guest for today's session, uh, Mrs. Vanshika Adiani ma'am, is not able to join us. However, until then, we will continue with the formal inaugural function. You are all requested to please stay connected with us. Very soon, we will start with the formal inaugural function. Until then, please stay connected with us. Thank you, Saurabh sir, for being understanding. Okay, so we may have lost internet connection with our honorable chief guest. However, we can still continue with our function because all of us we know that otherwise we are connected. It is the only barrier is this internet connection. So she may not be connected with us over the internet and in this meeting, but otherwise she is most certainly with us. So let us presume that it is at her hands that. We will be conducting this inaugural function. Let me please confirm whether my screen is visible or not, whether the presentation is visible or not.
Yes, your presentation is visible.
Okay,
Visit.
so we will now begin with the this thing uh, with the inaugural of the short-term training program. Okay, so that is where we declare that the 1 week short-term training program sponsored by All India Council for Technical Education and organized by Ballarpur Institute of Technology, Ballarpur, uh, on biotechnology and its application in tribal agriculture areas is hereby inaugurated. Let me check once again whether our honorable chief guest for the session is able to connect with us. Otherwise, we will proceed with our next session. Okay. I guess she's still facing the same difficulty connectivity issues. So now let me confirm. We can see that our honorable resource person for next session, Dr. Sanjeev Patankar sir, has already logged in. Am I audible to you, sir? Dr. Sanjeev Patankar sir?
Yes, you are on.
Sir, if you are ready, then we would like to continue with your session. No?
Definitely.
Thank you so much, sir. So, let's begin with our next session, and our honorable resource person for this session is Dr. Sanjeev Patankar sir. Dr. Sanjeev Patankar sir is PhD in microbiology from Nagpur University. He was earlier associate professor in Guru Nanak College of Science, Ballarpur from 1986 to 2014. He has also served as principal in Shiv Prasad Sadanand Jaiswal College of Gondia from 2014 to 2019. He has undertaken several projects that include minor research project of UGC on microbial production of glucose oxidase, young scientist project of DST on gluconic acid production by solid state fermentation, a major project of DST on fortification of tribal food from micronutrients by fermentation is also completed by him. Another major project of DST he has completed includes microbiological, phytochemical, and biochemical studies of asavas and aristas. Dr. Patankar sir has completed a project for Government of Kerala in development of spirulina on dicalcium phosphate waste. He has been a consultant for DME chemicals to establish biofertilizer plant and soil testing laboratory. He has published 20 papers in national and international journals. He is also an expert in Vidarbha Development Board, which is a statutory board of Government of Maharashtra for health and hygiene of tribals. Dr. Sanjeev Patankar sir is an academic counselor for Rashtrasanta Tukadoji Maharaj Nagpur University and board of studies member in Rashtrasanta Tukadoji Maharaj Nagpur University and also in Gondwana University. He is also president of Maharashtra State for Microbiologist Society of India. Well, I must say, Dr. Sanjeev Patankar sir, it is a matter of great honor for us to have you as our resource person for today's session. So, may I now request you to please start the session, sir.
Okay. So, good afternoon to all of you. And I am very much thankful to Ballarpur Institute of Technology for inviting me for such a lecture. And again, thank you for my lucid introduction. So, I am also thankful to Patron Sanjay Vasade sir, then Director of Ballarpur Institute of Technology. And now I will start my presentation. So I think this is the AICTE sponsored short-term training program, and its theme is on biotechnological applications in tribal agriculture. So now this is a training program mostly according to my perception is that for engineering faculty members, and the topic it is from biotechnology, and that too for the tribal area and for the tribal agriculture. So it's a very diversified type of the topic, and also the diversified audience also. So I will try my level best to make it feasible to understand the topic, and also you will enjoy the lecture. So may I start with the presentation now? So I will start with so is it visible?
So, right now we can see your screen. We cannot see the presentation.
Okay. Is it visible now?
Yes, sir.
Okay. So, today my topic, it is role of fermentation in treatment of tribal malnutrition, a case study for Gadchiroli district. Now, as I told you, this is a slightly different type of the topic, but related to what? Related to the tribal health and also the in some way to the tribal agriculture as such. So here, the very first thing, as it is what? It is a topic, it is what for malnutrition. So naturally, here the key words such you can see here, it is what is the tribal, it is the first is the malnutrition, then tribal malnutrition, and then the fermentation. So all these areas, they are as such, they are new. So we will go for in a brief way what are these things in a in this presentation. So nutrition as such, it is what? It is a nourishment, all of you know, or it is an element which refers to the nurturing of our body, which keep it healthy and functioning as it is supposed to do. Now, nature as such, it has provided the variety of foods. Now, all of you know that the people, they are using different type of the variety of kinds of food. Some may be vegetarian, some may be non-vegetarian, but nature has given a variety of foods as such, and much diversity it is there. And as per the diversity in the population of human being, the diversity in the nutrition is also seen. Now here, when we say the nutrition, what nutrition gives what? The nutrition as such, and why it is needed. Now all of you know, might be knowing that if we consider our basic unit of our life, it is what? Or the body, it is what? It is the cell. And this cell, it is made up of what? It is made up of the carbon, nitrogen, phosphorus, sulfur, oxygen as such. Now here, the main constituents, they are what? They are the carbon and nitrogen. Now, if you are getting the carbon and nitrogen, we cannot take the carbon and nitrogen directly from the atmosphere. So we have to take in some form. And what is that form? We take the carbohydrate. What is it? It is the organic matter. If we see the carbon dioxide in the atmosphere, it is what? It is the inorganic matter. But in both the cases, the carbon is there. But our cell also needs what? Our cell also needs the carbon, nitrogen, but in the organic form. So what we do, we take all these things in a certain type of the organic form, like carbohydrate, protein. It is a source of nitrogen. Then there is what is the phosphorus we require. Then we need the sulfur, and there are so many things as such we need. Now, how they are coming? Apart from that, there are various other such things or other such components or other such factors we need in order to have our body in the healthy position. Now, if we say that from where we are getting it from, commonly we say it is from the nutrition. And what it provide, it is what? It is the nutrient. So the nutrients are available and they are present in the different form, as I already said. Now, because of this consumption of nutrients or the food, we maintain our health, we grow, we develop a resistance against the infection, and so on. So this is a very complex type of the process, and this entire process, it is studied in one particular head that is called as the nutrition. So what is the requirement of our body, what is the requirement of our cell, and how it is utilized, that is what, and how our body get benefited, that is nothing but what? That is nothing but the nutrition. Now this nutrition, as a science, it was founded by Lavoisier, who is the father of chemistry. Naturally, as I told you, the nutrients or the basic nutritional requirement of our body, it is in terms of carbon, nitrogen, and the sulfur and the phosphorus and so on. So again, the chemistry occur, and that's why the Lavoisier, he realized that we are getting what? We are getting all these nutrients in some or other form, and that is what? That is mainly the organic form, and some are inorganic forms also. So Lavoisier, a great chemist, he studied this particular type, he studied this entire process, and then he gave the name to the science, how these particular chemicals, they are applicable for the betterment of our life, and that is what? That is nothing but the nutrition. So the science of nutrition, it is one of the youngest of the sciences. So that is what, that is the nutrition. Now, if we define the nutrition, it is what? It is nothing but the science that interprets the nutrients and other substances in the food in relation to the maintenance, reproduction, health, disease of an individual. So it is what? It is the interpretation of nutrient, means what? How the carbohydrate, how the nitrogen based compound, that is what, the protein, and then the other things, how they are what? How they are used in what? In the maintenance of what? Maintenance of our health. So because of which we can grow, we can reproduce, our health is maintained, and we give the resistance against the disease. So this is what, this is the interpretation of all these nutrients, how they are utilized in this function, and that is nothing but what? That is nothing but the nutrition. Now another term we come across, it is the diet. Many more time we use the nutrition and diet as synonyms, but there is a slight difference. There is a hairline gap between what? Between the nutrition and what? Nutrition and the diet. Now what is the diet? It is nothing but it is a sum of the food which is consumed by a person. Now it may be in any form. It may be the vegetable form, a vegetarian form, it may be a non-vegetarian, it may be a mixture, and whatever it may be. If you go to the back dates as such, if you go to the ancient time, when we are in what? When we are in the primitive stage, the man was in primitive state. At that time, he was also using the food. He was also using the nutrition, but that the form was different. It was the raw type of the food. Now we have changed this. Now we have what we have changed. We have not changed the nutrition, because at that time also they are using the carbohydrate. They are using they are using the carbon source. They are using the nitrogen. They are using the vitamins and so many other things. The things were same, but we had changed the form. And what is that change form? It is nothing. It is nothing but a diet. Now our diet, it is what? It is nothing but the daily sum of food which we consume as such, in order to what? In order to give the specific intake of nutrition or the nutrients which are required for the health. Because we need certain amount of certain vitamin, we need certain amount of the carbon, we need certain amount of the carbohydrate, we need certain amount of what? The protein, so that the nitrogen based components in our cell can be developed, like what? The amino acids, then the nucleic acids, and so on. So that is what? That is nothing but a diet. So it is the some of the food consumed which is by a person, which gives a particular type of nutrition necessary for what? Maintenance of our health. So that is called as a diet. So nutrition, it is what? It is a general term. Now what is health? Because we are much more concerned. Why we take the nutrition? Mainly for maintenance of our health, and then protection against the diseases. So what is health? Now let us see what is health according to the, it is a definition according to the World Health Organization. Health is a state of complete physical, mental, and social well-being, and not merely the absence of disease or infirmity. So this is what? This is the health. Again, we see it is the physical, mental, and social well-being. So all these things, it includes the social well-being also. Simply you are free from disease, it doesn't make that you are healthy, but you must be physically fit, you must be mentally fit, and your social status must also be proper, and it must be substantial. So that is what? That is nothing but health, diet, and nutrition. So what is the relation between what? Between the nutrition and the health? Now, as I told you, that nutrition, it is required for what? For maintenance of the health. Now how? Now as we say that always our development or someone's health, it is what? It is his birthright, or it is what? It is inherited by someone. Its mental ability, then again certain other activities, they are what? They are inherited from the ancestors. Now it is what? When we say the inheritance, it means what? It is the genetically related. Now how this genetic come in the way, and how the nutrition, it helps to regulate the genetic problem. Now the gene information, it gives what? The gene information, it gives our body constituent, main constituent, that is the protein. What is the information which is present in the gene, it is given by the protein. So the proteins must be synthesized, and for that purpose we need what? We need the carbon and nitrogen, we need the amino acid. So if our nutrition is not proper, the basic components like amino acids which are required for the build-up of protein, they will not be there, and your abilities cannot be expressed, because there are so many type of the things, the enzymes, hormones, so many things, they are what? They are related to your genetic potential. So if your nutrition is not proper, though the genetic potential exists in you, it may not be reflected, or it may not be expressed out because the fuel for that, it is not available, which is taken from the nutrient. Maintenance of structural integrity and the functional efficiency of the body tissues necessary for an active and productive use. So that is what is required. Then for your mental well-being, it is required. Why? Because mental cells or the nerve cells, they require what? They require the nutrient. Then ability to withstand the inevitable processes of aging with minimal disability and functional impairment. Suppose if your nutrition is very good type at your childhood, then no doubt the aging, it is inevitable. But as the aging occur, there are certain disabilities may come. So we have to minimize these disabilities, and they depend on the nutrition what you took at your childhood and how you are continuing. So that is how the nutrition is very, very important, even in your old age. Then ability to combat the diseases such as resisting to the infection, that is immunocompetent development, preventing the onset of degenerative diseases, and resisting the environmental toxins and the pollutants. We are much more exposed to the different pollutants, so resistance must be developed for that, and for that the nutrients are required. Now, nutrition and diet, they are correlated with health and survival among the humans in India from Vedic period. The rulers were concerned to achieve the food security for their deprived population. So they were always concerned that how their population remain unaffected from the hunger. But condition of the lack of availability of food for good health persists even in 21st century. So it is the reality that there is a lack of availability of food, lack of nutrients are there, and then it gives a general condition, a clinical condition, which we call as the malnutrition. And how you can detect a malnutrition? When you say the malnutrition is going on, there are certain characteristics, and they are characterized. There are markers of malnutrition, and the two main markers of this are anemia and the anemic condition that is less in hemoglobin percentage, and your low BMI, that is the body mass index. Now here, what is body mass index? Now body mass index, it is it is a parameter by which you can check whether a person is deficient in nutrition, whether he is malnourished or he is healthier one. What is this BMI, body mass index? It is nothing but it is a ratio. It is the ratio between what? It is the ratio between the mass of the body, which is in the kg. It is the mass of the body in kg divided by what? Divided by height in meters. The square of height in meters. So that is mass of body in kg divided by height square in in the meters. So that will give you the ratio. It will give you it will give the body mass index. Now if the body mass index, if this ratio of a particular person it is below 18.5, then the person said to be the underweight. He is the underweight, or it is called as a low BMI. So he is malnourished. If it is between 18.5 to 24.9, then the health is good and it is a normal type. But if it crosses 25, value crosses 25, then we can call the person is what? The overweight. So these are what? Now see, the overweight is also not a good sign. It is not a sign of good health. And the lower BMI or the low weight, it is not also the sign of good health. In both the cases, the person shows certain type of the problem. So there must be a balanced type of what? The BMI, that is at 18.5 to 24.9. Now, the malnutrition. Why malnutrition take place? It may be because of, now before going to the malnutrition, we must see what are the nutrients we take. Now in the last slide I have given you what is nutrition, and I said that we need what? Carbohydrate, protein, and certain other thing. So the carbohydrate, proteins, now they are called as what? They are called as the macronutrients. And if the macronutrients are lacking, then there are certain type of the condition that is called as the marasmus, kwashiorkor. Then the protein energy malnutrition means what? If the proteins are not there, when the proteins are not there, your genetic expression may not be proper. Energy, it is what? Derived from the carbohydrates. So the person may not have the proper energy. So there will be no proper type of the work potential and the physical potential. So that is called as, in general, the protein energy malnutrition. In both the diseases, there is what? There is a less amount of protein. Because of that, the mental ability and the physical ability both are what? Both are going down. And then the second, it is type of nutrient, it is what? The micronutrients. Now what are these micronutrients? Which are required in very small amounts, but they are very much necessary in order to process these macronutrients. And what they are nothing but the vitamins. All type of vitamins, you know, nowadays during the COVID, people, they are knowing the importance of vitamin, and they take the vitamin C, vitamin D, and they also take the zinc and all other type of the thing. So all these are what? These are the micronutrients. They are required in the small amount. Now their deficiency may be there, and because of that also the anemic condition persists. Now what happened? If we consider the two parts of the society, one is the affluent people, and another is what? Another is the very poor people. Now among the poor people, we have a picture of what? The protein energy malnutrition, that is what? Lack of macronutrient. And they show a lean, thin type of what? The body stature. And again, in case of what? In case of the affluent people, they may also show certain type of what? Certain type of the elements. And what are those? They have certain things, like they may have a night blindness or certain type of the such type of the things as such. They may have the specs and all other thing. Their vision may be impaired, and so many other things they are occurring. Now they are because of what? They are because of the micronutrient deficiencies. Now in the affluent people, they get the macronutrient, they eat macronutrient very properly, but they lose, or they are not using what? They are not using this micronutrient, and that's why such type of the deficiency, it is called as the hidden hunger. It is, hunger is there, but it is hidden, it cannot be expressed. Whereas in case of what? In case of the previous one, that is where the lack of macronutrients are there, we have the hunger is seen, hunger is expressed. You can see that the person, they are hungry because they are not getting the bread and butter. So, but these people, on the other hand, the second type, they are getting all the things, the carbohydrate they are getting, protein they are getting, but they are not taking the vitamin, the minerals, and so many other things. So apparently they are not seen to be the hungry, but they have the hunger for what? For these vitamins. So it is called as the hidden hunger. Nowadays, the major problem, it is because of what? It is not because of the macronutrient, but because of this micronutrient. They show the hidden hunger due to what? Due to the change in lifestyle, and there are so many other things as such. So we are having so this is what this is the now why this malnutrition occur? Now there are multifactorial etiology, means there are different number of factors they are responsible. Etiology means the reasons. Etiology we call as the reason of a particular disease or a condition. So there are multifactorial type of the etiology that is available. Now why it happen? It is because of what? It is the interaction of different things. On one hand it is the poverty, on other hand it is the social and cultural factors and other things. Now when we see the poverty, at that time the food inadequacy is there, the poor hygiene conditions are there, infection because of that occur, illiteracy and the lack. Now as the poor people are there, they may have the lack of education, so they become what? They become illiterate, lack of awareness of nutrition, lack of awareness it is there, no proper utilization of healthcare facilities, etc. And then they suffer from the different type of disease. As the disease condition occur, they are not properly nourished, so it becomes a vicious circle, that how the poverty gives the disease, how the poverty gives the protein energy malnutrition, and again they remain in the same type of the cycle for malnourished condition. Now there is another way, there is the social and cultural things, strong food beliefs and taboos, superstitions. Now these are what? These are available still in what? Still in the society, that this food should be taken, this food should not be taken, not only in the illiterate people, tribal people, but also in the affluent people also. Then early marriages of the girls, because of the early marriages of the girls, the adolescent pregnancies occur, and then they are malnourished, the anemic condition occur. And then the very important thing is that nowadays the changing lifestyle of the woman that occur. Now the changing lifestyle, it include what? The many more I am seeing here, many more lady people, female, they are what? They are employed. And as they get employed outside, they earn money, they have the education. So education on other hand also responsible, because what? The education gives what? It allows, or it pushes the lady for what? For outside of the home, and then the lifestyle changes. Then we are not taking the proper type of the food, we are taking the fast food and so many other things, easy to cook and other things. So because of that, certain vitamin, then the certain nutrients they are lacking. So generally because of such thing, nowadays we are facing the micronutrient deficiencies. That is what? That is the hidden hunger. So social, cultural and poverty, that is the poverty. So now there is an interaction between them, and we get what? We get the malnutrition. So now we are coming to the micro. Now as a whole, in my topic, or the title of my topic says that the fermentation, the role of fermentation in the treatment of the malnutrition among the tribal. Now this is what? This is one of the project, my project, funded by Ministry of Science and Technology. And here in many states, the malnutrition among the tribal, malnutrition among the rural people, it was evident. Malnutrition in the different sectors of, or in the different sections of society are there. And the tribal malnutrition, it was much talked, particularly in Maharashtra. There are two regions. One is the Melghat region, might be knowing, in the Amravati district. And then another is what? Another, it is what? It is in the, there are three regions, more or less, 3 to 4 regions where the malnutrition occur among the tribal. One is in the central Maharashtra, in the North Maharashtra. There is what? There is the Nandurbar district, between what? Between the or nearby to the Gujarat border. So there also the tribal, they suffer from the malnutrition. Then another is what? Another is the Gadchiroli district, where the Bhamragarh and the other area. And also in the Chandrapur district, we had seen the different races of tribals they are staying. Out of that, one is what? One is the Madia tribe. They are staying in what? They are staying in the Bhamragarh. That is what? That is in the Gadchiroli region. It is a dense forest, and then they are suffering from the malnutrition. So as our college, it was located, or it is located as such, in what? In the Chandrapur district at Ballarpur, which is nearer to the Bhamragarh or nearer to the Gadchiroli district. We took this particular topic, and then we have studied the nutritional status of the tribal, and then what they are eating. They are eating a traditional food that is called as, it's a fermented food that is called as the ambil, which is prepared from what? Which is prepared from the fermented rice, or there are certain wild grains which they grow and then they consume that particular fermented food. And after that, what is their health status that we have studied, and then we tried to give a certain type of the solution by a fermentation process. So for that purpose, we must see what is the nutritional status of the traditional food which they are using. They are not consuming any other food. This ambil or this tribal fermented food, it is what it is, their staple. Then they are not ready to accept any type of the help, any type of the counseling. They are illiterate. Acceptance, the social acceptance is very less. So there are different type of the beliefs and taboos among them. They are illiterate. So all type of the factors in the last slide which we have seen, the poverty and the social economic problem and the social culture, social and cultural problem, all they have culminated and they have given the malnutrition. So let us see what is the composition. This is the these are the people. Now you can see this is the typical Madia who is in the primitive state. Now they are staying in the deep forest, in the group of what? In the group of hutments. They are called as the tola. Now what we did, now this is the health feature of Madia tribe. Now what they show the anemic condition. Reason is what? Because of folic acid deficiency, which is the vitamin which is necessary for development of hemoglobin. Then they also have the chronic malaria, low body mass index in the adults, and then the infestation with what? Intestinal diseases and so on. Now what we did here. Now these are the wild grains used in the ambil preparation. Now apart from the rice, they use such type of the grains which we have not come across in our daily routine life, or they are not available in the urban areas also. So harak, mande, kutki, these are the wild grains which they grow by shifting agriculture. Now what we did, we collected the ambil sample. We divide this Gadchiroli district in 3 parts on the geographical basis, and nearly about 200 samples we have collected, and then we process them to give the proximate composition. What is the proximate composition? It gives what? It gives the percentage of what? The percentage of protein, carbohydrate, fats, energy as such, and then how much amount of water is there. Apart from that, we did the micronutrient analysis also. That is what? Which include the vitamin B, different type of vitamin B complex, B1, B2, B12, folic acid, and so on. Apart from that, certain type of minerals which are very much necessary, zinc, iron, calcium, and so on. So at the same time, when we were collecting the sample like from these families, we have collected the blood samples also from 1 adult male and 1 female from each. And then we had done the hemoglobin percentage also, and we took the BMI of adult male and female from that family. So we got the data regarding their BMI and the hemoglobin percent, and then we tried to correlate by statistical methods that how the food deficiency it can be correlated, or how the vitamin deficiency can be correlated with the health status. Now let us see what we have got. This is the proximate composition. So 100 samples we have analyzed, and what we did all the standard deviation and all other things. So this gives what? This gives the moisture, proteins, fats, ash, crude fiber, carbohydrates, and then the energy which is available. Then B complex vitamin per 100 gram of the ambil, or on the dry weight basis. So niacin, folic acid, cyanocobalamin, that is vitamin B12, riboflavin, vitamin B2, thiamine, B1. So we had done. Then iron, sodium, potassium, zinc, and calcium. So this is what the trace elements. See, so these are what? Vitamins, and then the trace element, they are the micronutrients. Previous slide which was showing the protein, carbohydrate, energy, and all other things, they were the macronutrients. Now we saw the health status of Madhya tribes. Now, what was that? Now, this is the 70% of the male. They are what? They are below normal level of hemoglobin. In male, generally, we must have the 11 to 12%. It is the ideal one. Below that, they are what? They are low in hemoglobin. Whereas in female also, we have the similar type of what? The hemoglobin, and below 10, they are what? They are the anemic. So among the male, 70% people, they are what? They are, or 70% male, they are anemic. The 59% female are what? Are anemic. Then high hemoglobin, nil, normal, you can see here. Then BMI, depending on the body mass index basis, underweight, normal, that we have seen. So underweight male, they are 70%, 42%, they are what? They are underweight. So overweight was no one. Now here, the nowadays also, if we look at what? At the recent analysis or recent survey which was done by the National Family and Health Survey, that is called as NFHS. Now NFHS, they have done the survey recently. The report has come. Now this is the tribal situation, but nowadays what we are getting in the urban population also, the anemic condition, it is nearly about, it is in 68.9% of what? The adults. Now this is what? This is the alarming situation. Now in case of what? In case of the adults also we are getting nowadays. Then in the rural area, the stunted children, they are also seen. At the same time, in urban area also we are having a similar type of the situation. And in that, one more dimension it has come, and that is what? That is the that is the overweight. That is the overweight situation among what? Among the children, that is going on. So some children, they become overweight. Now why they are overweight? Again, it is what? It is the lack of awareness regarding the nutrition. So that is again an alarming condition. So this is what? This is the percentage of overweights, they are also increasing, and the underweights, they are also increasing. Hemoglobin levels, they are also going down, not only in the rural but also in what? In the urban area also. This is the today's situation. This is the fresh survey 3 days back which has come, or which has been published, and this is the situation which we are seeing in what? In the tribal people, that is in the Gadchiroli district, which is in the very deep area. It is a Naxal affected, and so many other things are there. Now this is the percent deficiencies of nutrients regarding niacin, folic acid, vitamin B12, B2, iron, sodium, potassium, and calcium. Now you can see, now we have calculated RDA. It is what? It is the recommended dietary allowance. Recommended dietary allowance, it is what? Is the amount of a particular vitamin should be taken per day basis. This is recommended by Indian Council of Medical Research. It is 15 milligram. Now we have calculated daily intake or intake per day by these tribes. On dry weight basis, they are consuming nearly about 500 grams of the ambil per day. Accordingly, we have calculated the deficiencies, how much vitamin it is going inside through the ambil. So this is what? This is the daily intake, and the differences are like this. And the deficiencies you can see here. For niacin, it is 95.6. Folic acid, it is 98. Vitamin B12, it is 62.8. Vitamin B2, it is 99.8. Iron, 45.1. Sodium, potassium, and the calcium. Now these are what? These are the deficiencies which are present due to what? Due to the ambil consumption, and that is the staple they are using only. So this is what? This is the antimicrobial activity by the ambil which has been shown. That is, it may act against what? Again, the Staphylococcus aureus, which causes the food poisoning effect. It can act against the typhoid disease. It can act against the E. coli. It can act against the Proteus vulgaris. So now what are the different type of organisms which we have isolated? Now there are certain bad organisms and the good organism. Out of that, the E. coli we are getting in the 60% samples. When we get the E. coli, means what? It is the indicator organism. It indicates that there is a high contamination of enteric organism and the enteric pathogens. Bacillus, it is in the 20%. Lactobacillus is a good organism. Micrococcus, it may cause certain diseases. Leuconostoc, again it is a good organism. And there are certain yeast and mold we are getting. So yeast and mold, they, particularly the mold, like fungus, they may give certain type of the diseases and certain type of the pathological conditions as such. Now we have gone for the seasonal variation. How many standard plate counts? That is what the overall microbial population, which is expressed in 3.9 into 10 to the power 7 number per ml. This is what this is in the month of December. Then in the May, July. So you can see that in the May we get the highest number. Now there will be a surprising question when the heat is very high, the temperatures are very much high, how they are surviving. The reason is that these tribals, they are preparing this food in what? In the earthen pots, that is matka. And there what they do, they ferment it by adding the water. Inside the earthen pot or in the matka, the temperature remains 30 degrees, where the organism grow very nicely, and it will not be damaged or it will not be dead because of the high temperatures which are present on the outside. Then in the December, coliform plate count, that is like E. coli, their count go on increasing. Their count it is less. Again in the May we get the higher one. Then yeast and mold count, again there is a similar type of the trend we have seen. So this is what, this is the seasonal variation in the microbial flora which is present. So in all we can say that the the umbil is highly contaminated. So unhygienic conditions are there. So this is what, this is our conclusion. Now this is what age-wise hemoglobin in the males. Now you can see here, they between the age group of 18 to 27, it is 10.84. That is what, it is below normal. So they are anemic. As they grow 28 to 37, again it decreases. Why? Because after 18 years they start work, they do certain work, they go for shifting agriculture and so many other things. As they grow further, up to the 48 to 57, their hemoglobin goes down. After 58, it may increase slightly because they stop their working. They are not that much fit, but their hemoglobin slightly increases because of what? As they become the adult, they become the owner of the family, so they get nice or they get some substantial amount of food as such, because of what? Because of the general rule that exists in the families. Then age-wise hemoglobin in the female, it is lower, 9.71, 8.70. As between the group of 28 to 37, early marriages are there, pregnancies are there, loss of blood, loss of weight, so many things are going on. Up to 38 to 47, again still it goes down, and then at the end again it goes very poor. That is what, up to 6.20. That is the worst condition that we have ever seen. Now this is what, when we did our analysis, as I told you, we were taking what? We are taking the samples of this umbil, that is the fermented food, and we are also doing the hemoglobin percentage in what? In the male and female. So what we found, there are certain umbil sample where the folic acid levels are very good in some families, and we compared that the hemoglobin levels are comparatively better than the others. So we sorted all such type of the sample, and then we tried to get the correlation analysis. A correlation analysis is a statistical analysis, and there we get a positive relationship indicating what? As the folic acid content go on increasing, in what? In the umbil sample, the hemoglobin percentage also go on increasing. So that is what, there is a positive relationship that we get. So we could conclude that if the folic acid, it is given in a proper way, if we increase the folic acid content in the umbil, then the hemoglobin levels can be increased, and then the anemic condition can be addressed. So how we can improve that? because it is a fermented food. Now, thing is that they are not taking any other type of the treatment given by the government. For example, the folic acid and iron tablets, they refuse to take. They are not taking the fortified atta, iron fortified atta, and so many other things. They are illiterate, very poor, and in the primitive condition. So what we did, we tried to uh, improve the fermentation condition. We tried to give a new method for this ambil preparation, which is workable at their home levels, and then we could transfer that particular thing to these people, so that they can improve their hemoglobin levels, health status, without changing their food habits as such. So what was that particular effort? So in conclusion, we can say that overall population is anemic irrespective of sex. Among female, the average hemoglobin range, it is between 6.2 to 9.7, so it is very less. Among male, it is 8.8 to 10.8. Average survival age in both the sexes is 45 to 50. In few cases, it may extend up to 70. A correlation in the hemoglobin and folic acid content of ambil exists. Families showing higher hemoglobin percentage show higher folic acid level in their preparation of ambil. According to government surveys, the child mortality in this region is very high due to malnutrition. This is because the parents, particularly mothers, are anemic and malnourished. There is an urgent need to improve the health status of mothers and in general the parents. So we have to look for the parents also, because if the parents are not healthy, the child is not also healthy. So previously what people are focusing only on the child, but they are not considering their mother and father. But now we have considered here both the parents and mothers and also the child. So these are our conclusion. Now we had tried our fermentation process. Now let you know what is fermentation. Fermentation, it was coined by Louis Pasteur. Classical definition: conversion of sugar to alcohol by yeast under anaerobic condition. Now this was the classical definition which was given by the Louis Pasteur. Because what happened in the European countries, they are preparing the wine. They prepare the wine at home. How they prepare? They take the grape juice, add the yeast, cover it. When you cover it, it is cut from the oxygen. So because of that, it gives what? It gives the anaerobic condition, and then the alcohol production occur. So conversion of sugar to alcohol by yeast under anaerobic condition means absence of oxygen. That was the definition given by Louis Pasteur. Fermentation, it is the word which is derived from the French word, that is what to ferment means to boil. Now what happened here? How the boiling comes? When the fermentation is going on, there is an evolution of carbon dioxide, and it gives what? It gives a vigorous bubbling. So that bubbling, it gives the look of boiling. So that's why it is called as the fermentation. Now the biochemical definition was given as the science progressed: a reductive process in which large organic molecule is converted into smaller molecule under anaerobic condition. Now this is what the large molecule means. What? The sugar, it is the, say, if it is the sucrose, or if you say the glucose, C6H12O6. Now there we get the alcohol C2H5OH. So it is reduced, or its larger molecule is converted into the smaller molecule under the anaerobic condition. That is a reductive process. So this is the biochemical definition which was given. Now the fermentation, it has got different uses. Now it gives different type of biochemical solvent, acids. It gives the bioenergy like bioethanol, biogas, biocells, then biomass, spirulina, then yeast production, then the enzymes and therapeutics. In the food, the traditional tribal foods are there, then traditional tribal foods are there, then traditional urban foods which include the curd, shrikhand, and so many other things. Among the tribal, it is the ambil. Then there is the pickles which are also there. Some Indian fermented foods, all of you know, that is the idli, dhokla, then bhatura. These are what? These are the fermented food. Now there are oriental food also. All of you know the soy sauce, Many more time we consume this soy sauce nowadays in the Chinese food, then the sufu and miso and so many other things. So these are what all these include the fermentation, all these fermentative processes, and fermentation has got different applications. Now, how we developed the particular type of what? Particular type of the improved method and the fortified type of the ambil for this tribal. Now this is a symbiotic approach in fortification of vitamin B complex levels of traditional tribal food ambil in Madhya tribes of Gadchiroli district. Now the symbiotic, it is not the symbiotic, it is a symbiotic, it is a different term. And what is that? Let me know. It is nothing but it is derived from, or it is a mixture of two things, that the probiotic and the prebiotic. Now, what is the probiotic? It is a live microbial feed supplement which beneficially affect the host animal by improvising its intestinal microbial balance. So you take certain type of the live microbial feed. For example, many more time you come across the advertisement of one particular product that is called the Yakult. What is this Yakult? It is nothing but a microbial culture. You come across the yogurt. What is this? It contains the live microorganism which improves your intestinal microbial flora and which gives the beneficial activity. So that's why it is called as a probiotic. So that is what. Now what is the mechanism? These probiotic organisms, they produce the antimicrobial substances which gives the competition for nutrients and the pathogens. They compete for adhesion with receptors. They stimulate your immunity. So they protect your body surfaces, intestinal surfaces from different pathogens. Beneficial effects which are shown: non-infectious dysbacteriosis after the high dosage of antibiotic and after the radiation therapy. Many more time you come across doctor prescribe you certain antibiotic, at the same time he gives you a another supplement of vitamins. So vitamin B complex tablet. Why it is so? Because your vitamins are vitamin B synthesizing flora of your intestine get destroyed by antibiotic. Now because of that, the other organism attack on your intestine, and then you suffer from the diarrhea and other things. But suppose if you take the probiotic culture along with the antibiotic, you may not suffer from this and you may not have to require the vitamin B complex tablets. So that is the use. Then health promotion, inhibition of carcinogenesis, that is, it prevent the cancer formation. Anti-cholesteremic means what? It doesn't allow the increase of cholesterol levels, which is a much more problem among the affluent people and also in the rural people. High calcium resorption is there. Destruction of anti-nutritional factors. Anti-nutritional factors, these are what? These are the certain factors which are present in your food which doesn't allow the digestion of a certain type of the factor, certain type of the nutrient. For example, in the legume plant, there is what? There is one anti-nutritional factor, phytate, which doesn't allow the digestion of what? Digestion of the protein which is present, because that phytate, it doesn't allow the action of the enzyme which is present inside our intestine that digests the protein. And that's why always such type of the grains, or such type of the things we have to take only after the proper cooking. If you take them raw, they will affect. During the cooking, such type of the factors, they are destroyed and then they are easy to digest. So vitamin synthesis, riboflavin, vitamin B12, folic acid, pyridoxine, niacin, and the protein predigestion. Now possible mechanisms: improved protein metabolism, improved vitamin metabolism, prevention of constipation. Now there is a constipation is a general problem. So many advertisements they are coming in the television, that how you can prevent the constipation. Very simple. If you take the probiotic culture, organic acids which are produced by them, it will increase your intestinal peristalsis and give the normal bowel movement. Then antibiotic production, again different pathogen. Treatment in the liver damage, the probiotic preparations, they are very much useful. Reduction in what? Reduction in what? The colon cancer. So that is a very important thing. It improves your immune system. It initiates the production of IgA. in immunoglobulin A. So this is very, very important. Then lactic cultures, they are showing immunomodulatory effect. Lactic cultures, they are very much useful in synthesizing what? Synthesizing vitamin B12 and so many other things. Then the effect of ingested lactic bacteria start in the gut associated lymphoidal tissue. It increases your immunoglobulin, that is one type of antibody level. Lowering of cholesterol level, I told you. Now these cultures, they are mostly the lactic cultures, which are lactic cultures are mostly used in curd preparations, like, and then the yogurt preparation. They include these type of the culture like lactic Lactobacillus casei, Lactobacillus acidophilus, Rhamnosus, Lactobacillus delubrecae, and Bifidobacterium. Now, what is prebiotic? Because synbiotic is a mixture of probiotic and prebiotic. Now, probiotic include these organisms which I have given. Now, prebiotic, it is what? It is a non-digestible food by human system, but it beneficially affect the host. How it beneficially affect the host? It is non-digestible by the human, but still it affect. How? Because it stimulates the growth of the probiotic organism. That is very important. So as the probiotic organism grow, it allow the probiotic organism to grow, and it inhibit what? It inhibit the different other bacteria. So because of that, probiotic organism grow, and it gives what? It gives the improvisation of your health. So if you want to grow a probiotic, you must have to take the prebiotic. That is such type of the component which will help for the promotion of the probiotic growth. So quality is required for food ingredients to become prebiotic. They are what? They are the, it must be the neither hydrolyzed nor absorbed in the upper part of the gastrointestinal tract, GI tract. Be a selective substrate for one or limited number of beneficial organisms. Consequently, be able to alter the colonic flora in favor of healthier composition. So it will induce the luminal and systemic effect. So it will give what? It will give proper type of the effect as such in what? In your. Now what is that? All non-digestible foods are not prebiotics. Now keep in mind, all prebiotics, they are the non-digestible foods, but all non-digestible foods are not prebiotics. So which are the prebiotic food? Now here, resistant starch, it is a colon food, but it is not a prebiotic. Starch, it is not non-starch, it is not a prebiotic food. Hemicellulose, it is also non-digestible, it is not a prebiotic food. Pectin, it is also not a prebiotic. But what is that? The inulin. Inulin, it is what? It is a difructose. That is what it is a dimer or a polymer of fructose. That is called the inulin. Where it is present? Inulin is present in different component, different type of the food, including banana, then the onion, and so many other things. So they are what? That is the prebiotic food. So sources of inulin. It is the onion, then the chicory, asparagus, banana, rye, Jerusalem articles, dandelions, and so many other things. Apart from that, we find the inulin and the prebiotic type of the quality. It is also present in the tribal grains, which I have shown earlier. That is what the harak, mande, kutki, and so many other things which I have shown, which are not common in the urban population. So synbiotic, it is what? It may be defined as a mixture of prebiotic, probiotic that beneficially affect the host by improving survival and implantation of live microbial dietary supplement in the gastrointestinal tract by selectively stimulating the growth and or by activating the metabolism of one or limited number of health promoting bacteria, thus improving the host welfare. So that is what there is a synbiotic. Now we have isolated certain organisms from the umbil itself, which are very good. They are health promoting. Now we have isolated one bacillus species. It was isolated and identified as Bacillus pumilus, which produce what? Which produce the vitamin B12, which produce also one antibiotic, that is pumilin. And we had deposited it at National Collection of Industrial Microorganism, and we got the accession for that. Then another is the leuconostoc species. It is the leuconostoc mesenteroides which developed what? Which developed the dextran. A gum is there, and it is deposited at National Collection of Industrial Microorganism at Pune. Accession is NCIM 5200. Now, apart from that, we have other cultures, lactic cultures, which include Lactobacillus, Pediococcus, leuconostoc on the basis of their tests, biochemical tests, and the sugar fermentation. Now, we had prepared different cultures, and we tried to prepare the ambil in the conical flask at our laboratory. So we have used these pure cultures, leuconostoc species, Bacillus pumilus, Lactobacillus. Then we have mixed LEBS, that is what? The leuconostoc, Bacillus pumilus, and then the Bacillus sporogenus, Lactobacillus sporogenus. Other cultures, we have indigenously isolated lactic cultures, lactic 1, which has got the equal parts of the lactic culture DC, BA, BD, lactic 2, equal parts of. Now these are the all lactic cultures we have isolated from what? From the ambil. Then we have obtained the curd from market, and then we are used for that. So we have these options from where we prepared what? We prepared ambil, and then we again went for what? The proximate analysis of the ambil product, vitamin B levels, and then the micronutrient level, that is what? The trace elements also. So we have standardized the parameters like moisture 80%, temperature 37, time of fermentation it is 16 to 20 hours, inoculum size, that is how much culture should be added on the basis of what? The amount of the raw material which we have taken, it is the 10%. And the raw materials were what? It is the 50% or the 5% of the rice and 5% of the kosri that we have taken. So now the standardization of inoculum, which inoculum is better? Now you can see here we got the lactic 1. It is showing what? It is showing a good sugar production. Then which inoculum shows the better protein production? If you use the curd, it gives the highest protein production between what? Between the 20 hours. Then after that, niacin production, again the lactic 1, you can see here it produces what? The niacin in a very good condition. Then folic acid, it is given by lactic 1 within 16 hours. How much amount it is giving? Very good kind of the folic acid it has been produced in the ambil. Riboflavin, so curd is giving, lactic 1 is giving. So these are what? These are the various things. Then now vitamin B12, Bacillus pumilus, it is giving what? It is giving highest quantity of what? Of the vitamin B12. Generally, all of you might be knowing vitamin B12, it is derived from the non-vegetarian food. And there is what? There is a lack of vitamin B12 deficiency or vitamin B12 or the vitamin B12 deficiency among what? Among the vegetarians. So if vegetarians are using such type of the probiotic where the vitamin B12 is increased, so you can have the better option. Then the thiamine, again the lactic 1, it is showing. Then the combination of what? The kosri and rice. The kosri is what? It is the wild grain. This is 5% quantity we have mixed in equal proportion. So there with the lactic 1, curd, and we are getting what? We are getting the highest in case of what? In case of the curd. Then niacin. Niacin again in the lactic 1 we are getting. Then after that the lactic 1 again folic acid. Then lactic 1 again it is giving the riboflavin, vitamin B12, lactic 1, curd, and these are what? These are the various type of the cultures there that we are getting. Now this is what? Lactic 1, lactic 2, and they are the thiamine production. Now following conclusions we got. Now here, if we blend other locally available grains with rice, can yield the ambil with better nutritional content, particularly folic acid and the cyanocobalamin, that is vitamin B12, shows higher values than ambil prepared from the rice alone. So if we suggest it to the tribal that you use like this, then we could do this. Preparation of ambil by using such blends may help in increasing hemoglobin level because folic acid is increased. Some important in the grains like kosri must have favoring a metabolic pathway of folic acid and cyanocobalamin, and it may contain what? It may contain a certain type of the prebiotic character. To ascertain there is an urgent need of phytochemical analysis of such locally available grains or the foodstuff. Tribals must be educated or trained to utilize such grains in a judicious combination and must use a proper type of inoculum for the umbil fermentation. Now, what we did, we went for simulation. We prepare similar condition as their hutment, and we prepared the umbil in the laboratory. So it has given again the different type of the results like this. Protein content, it has been increased. Niacin, then folate, it has increased. Then riboflavin, then this is what? This is the thiamine, and so many other things are there. Then this is what? This is the, we have used the two separate inocula. That is what we are using, the 5% PCR means what? It is the partially crushed rice, 5% kosri, lactic 1 inoculum. Then the average temperature, it is 34 degree. Fermentation is of 16 hours. Container, it is of earthen pot. Then P1, the conditions, they are similar, only dried pellets which we have prepared. They are containing what? They are containing the lactic 1 and 84B. Now this is a special type of the culture. So the pellets we have prepared, we have added what? We have added the leuconostoc culture. This leuconostoc culture, it gives the dextran, which is a gum, and because of that we could get such type of the pellet. We dried them. These pellets, there the organism, it gives the viability for 7 days even at room temperature. So that we are tested, and then what we did, we added those pellets in what? In the in the earthen pot, and the fermentation was allowed. So the idea was that we could prepare such pellets and transfer it to the tribal people. So once they are transferred to them, now that culture become there. Why? Because the practice of the tribal people is that they never wash their earthen pot for the next fermentation. So organism remain there and the fermentation is carried out. So this culture can be cultivated in a continuous manner as such inside their earthen pot likewise. Now this is what? This is the comparative study with initial level, and when we use the pellets. So you can find here the total proteins, they are increased by 36%, riboflavin by 81%, then cyanocobalamin by 66%, that is vitamin B12, folic acid by 59%, and thiamine, it is greater than 100%. So this is what? This is our way. Now conclusion, this is what? 79% increase observed in the riboflavin, 37 in the vitamin B12, more than 100% in the niacin, 59 increase observed in the folic acid, 100% increase it is in the thiamine. Bacillus pumilus is responsible for elevation of cyanocobalamin level. Bacillus pumilus also exhibited antimicrobial activity against the human pathogen like Staphylococcus aureus. It produced antibiotic, that is pumilin. 36% increase in the total protein content. So this is what? This is very important. Now this is the Madia tribe. We can see future scope. There is a further scope to achieve even higher levels of certain B complex vitamin by experimenting combinations of various other raw materials. Further work is still underway. Now, what efforts we have done for the implementation of these results? We submitted reports and suggestions about fortification of vitamin B complex in tribal food to Health Department of Maharashtra and requested for assistance for implementation. Submitted report to central government. Pellets were distributed among the tribals in Etapalli Tahsil of Gadchiroli district. Through NGO, shown increase in the hemoglobin levels, but no response from the state government. That is the pitiable thing. In March 2014, repeat surveys in the Bhamragat Tahsil was done through Vidarbha Development Board as a member of Committee on Health and Hygiene of Madia tribe. Report submitted to Honorable Governor of Maharashtra. The report is this. This is the report which I have submitted. Now, We are grateful for this work to Department of Science and Technology, New Delhi, National Institute of Nutrition, ICMR, Hyderabad, National Environmental Engineering Research Institute, CSIR, Nagpur, and this is what the laboratory where the work was carried out at Ballarpur, and this is the college where the entire project was carried out. So thank you. If there is any query, you can ask. Thank you once again.
The attending members are requested, in case if you have any queries, you can always unmute yourself and you can talk to our resource person directly in case if you have any queries. Or you can also type in your queries in the chat box and they can be answered. Until then, sir, I have a query. I suppose you have already covered it in the presentation. However, still I would like to, because I myself suffer from vitamin B12 deficiency.
Okay.
that is the reason.
Common thing. That is a common thing nowadays.
Yeah, it is very common, sir, but it took me a long time to understand as to
Right.
How fatal it can be, because it affects our central nervous system directly.
Right.
And I, being a pure vegetarian, I would like to understand as to what are all the things that we should try. There are two questions that I have.
Generally, it is a problem of vegetarian people, where vitamin B12, it is source is non-vegetarian. So better you take the curd, better you take the yogurt, you take the Yakult, and you could overcome it.
Okay, sir. And one more question, sir. In many patients, they say that though we take vitamin B12, it is not easily absorbed in the body. So for that, is there anything else that can be done?
Vitamin B12 means vitamin B12 tablets?
Yes, vitamin B12 tablets as well as vitamin D both. Vitamin D also.
Exactly what you are saying. See, all these vitamins, they are in the pure form. They show such type of the problem. Even I told in my presentation that tribals, they are not accepting the folic acid iron tablets. Why it is so? Because it gives the nausea. It gives certain type of the problem, and that's why they refuse to take. So in the natural form, if you take any type of the vitamin, that will be more better. So natural forms, it include what? It include vegetarian and non-vegetarian source. If you are vegetarian, you try to take as much as the curd, yogurt, and the Yakult. So that is what that is the thing.
Okay, sir. And one more query in the same direction. How about milk, sir? Is milk also a rich source of vitamin B12 or not?
Vitamin B, see, in the milk, the amount of vitamin B, any type of vitamin B, it is lower than what is in the curd or in any fermented food or in the fermented milk product. Because when these organisms, they grow, they synthesize it in a natural way. So, yeah.
Thank you so much, sir. So if any of the other attending members, in case if you have any queries, please go ahead and you can unmute yourself and ask your query, or you can type in your query in the chat box. Well, then I suppose that there are no more queries, sir.
Right.
So, sir, on behalf of Ballarpur Institute of Technology, Ballarpur, and on behalf of the organizing committee of this short-term training program, I thank you very much for conducting such a wonderful session. Also many thanks to you for joining us before time and adjusting to the schedule, because we lost our connectivity with our chief guest today, and that was the time we needed you very much, and you were there. Thank you very much for that too, sir. Thank you so much, sir.
Thank you. Thank you.
Now, to all the attending members, the link to submit the feedback of this session will be shared shortly on the WhatsApp group, and you are all requested to please fill in the feedback before 11 p.m. tonight, because that is after that the link you won't be accessible. So you are all requested to submit the feedback until then. And we will now take a short break from 1:45 p.m. to 2:30 p.m. as scheduled, and our next session will start at 2:30 p.m. Please do not miss that session. That session will be addressed by Dr. Radhika Markar madam, who happens to be a postdoctoral fellow in Institute of Chemical Technology, Mumbai, which is a very famous institute of India. I will request all you, in case if you wish to, in case if you wish to leave the meeting, you can do that now, but please do join back before 2:30 p.m. I will request all of you to join us back by 2:25 p.m. so that we will be able to start the session at 2:30 p.m. sharp. You can join back using this very same link. The link which you have used to join now, the same link can be used to join back after the recess as well. So thank you very much for connecting with us, and see you soon at 2:30 p.m. Thank you.